Hi everyone, we are again live in YouTube. So if you want to share the link, you can do it now. We have 5 more minutes and we will be back. Sorry.
Hi all, for your information, I have put the YouTube link on the chat. Thank you.
We return with the coffee break. We welcome again. Our next speaker is Prashant Sharma. He works at the University of Wisconsin in Madison. He's focusing in invertebrate macroevolution, and he will talk about ice loss in troglomorphic arachnids. His talk will be in English, and he can receive questions in English and maybe Spanish with some help. So thank you very much, Prashant. You can start.
All right, Alejandro, thank you so much. Quick questions. Can you see my screen okay?
Okay.
Yes, we can.
Okay, fantastic. All right, and so I'll go ahead and get started then. So one of the broad questions that my lab is pursuing is the process of morphological convergence. That is, how is it that cave adapted species convergently evolve troglomorphism? So caves are opportune locations for examining the genetic basis for morphological convergence, in that they present this well-defined suite of characteristics that we refer to as troglomorphism. That is the repeated observation that cave-adapted species will, for example, lose eyes, lose pigment, and undergo compensatory evolution of such structures as olfactory organs or non-visual sensory structures like tactile organs. So much of what we know about this conversion phenomenon is driven by two different model systems: the Mexican cavefish, Sionex mexicanus, and the European isopod, Acellus aquaticus, the latter being largely driven by work from Meredith Protas's lab. So while we've learned quite a bit about how it is that these lineages undergo adaptations to life in darkness, and principally a lot of that attention is focused on how it is that eyes are lost in caves, there's one group of animals about which we don't know very much with regard to the genetic basis for troglomorphism, and that is the arachnids. So arachnids, groups like spiders, scorpions, and mites, are important components of cave ecosystems, being among the notable predators in such environments. And the question as to how it is that arachnids have lost eyes in caves, much less how they develop, is largely unexplored. What we know from the cavefish and the isopod system is to some degree difficult to transfer to arachnids for two reasons. The first obstacle to our understanding of how it is that arachnids form their eyes is that the eyes of arachnids are structurally and functionally different from those of pancrustaceans, so groups like insects or isopods. So shown here on the left is the head of the fruit fly, Drosophila melanogaster. The large and main eyes that you see here are the compound eyes. So these are composed of little structures called ommatidia, and they have separately a smaller set of eyes at the top of the head called the ocelli. What you will notice in this arachnid is that it does not have compound eyes. Extant arachnids do not have compound eyes. They have instead a very different number and arrangement. So in this photograph, this large pair of eyes that we see here, these are called the median or primary eyes. So these are simple eyes, so ocelli. And in addition, we see a couple of lateral eyes. So here we see three pairs of, and these are divided into anterior, median, posterior lateral eyes. These are structurally and functionally different from each other as well. So principally what differentiates these two types of eyes is the location of the photoreceptor bodies with respect to the photoreceptor rhabdoms. So in the secondary eyes, the rhabdoms are proximal to the photoreceptor bodies. So this is an inverted eye type. The secondary or the lateral eye, it's a lot like the vertebrate eye. It's kind of like our eye. The opposite is true for the principal eye. Secondary eyes are also differentiated by the presence of, in some arachnid groups, of a reflective layer called a tapetum. So the other complexity is that in certain spider lineages, these secondary eyes are also structurally and functionally different from each other. So you can have a couple of different eye types with different tapetum types as well. The second obstacle to extrapolating from the isopod system is that a certain subset of arachnids that we call the arachnopulmonates, so arachnids that have book lungs, things like spiders and scorpions, share a duplication event, a putative whole genome duplication event that occurred more than about 450 million years ago. And as a result of this whole genome duplication, they share two copies of lots of genes that are involved in embryonic patterning. And this creates some complications. By contrast, arachnid groups like mites, ticks, or harvestmen will have a single copy of said genes. Okay, why does this matter? Shown here is a suite of genes that are important for establishing eyes in the fruit fly Drosophila melanogaster. So over the last half century, much of what we've learned about how eyes are formed in arthropods comes from this model organism. And As an example, if we were to misexpress, and this is one of these classic experiments that was done many years ago, if we were to misexpress homologs of PAC6 ectopically all over the fruit fly, the result is ectopic eyes that will form all over the body, for example, on the antennae or the legs or what have you. So you can think of these as very high level genes in the eye patterning network. The number of boxes that you're seeing here in this diagram shows you how many copies of these occur in the fruit fly genome. The connections between these genes is something that we refer to as the RDGN, the retinal determination gene network. I'll use RDGN throughout this talk. It refers to the gene network that is required for the establishment of eyes. Now, much of what we know about RDGN genes in arachnids is based entirely upon two spider species. One of them is Parasteatota tepidariorum. And it was shown 5 years ago that spiders have 2 copies of half of these genes. So as a result of that whole gene duplication, presumably, they've retained 2 copies of all these different genes. Beyond having 2 copies, one interesting phenomenon that we see is that these genes have subdivide, or rather, I should say, they exhibit spatial and temporally distinct expression domains. So typically, one copy is expressed after the other, and they are expressed in slightly different eyes. So in this figure from Schoenberg et al., the black arrowhead is showing you in an embryo. The dark shaded expression is the expression of a gene called cineoculus 1. Here it is in the median eyes. And the white arrowheads are showing the lateral eye primordia. The paralog, cineoculus 2, is expressed later in development, and it occurs only in the anterior lateral eyes. I'll summarize those dynamics using these kinds of cartoons. Again, these large discs here are going to be your median eyes, and these on the sides will be your lateral eyes. So in this schematic then is shown everything we really know about eye patterning in spiders. The 2 species that are shown here are the cobweb spider Parasteatota and the wandering Trachelaid spider Cupienius. And what you'll observe is that these 2 copies will often subdivide functions or have distinct expression patterns. And what we can also observe is that the expression patterns are slightly different from one species to the next. Every eye type in this diagram, every circle, is uniquely defined by some combination of transcripts, by some combination of colors. So this offers potentially a really elegant solution to how you make different eye types in spiders. There's just one problem. There are absolutely no functional data linking any of these expression patterns to any eye phenotypes in arachnids. We don't actually know what any of these genes do, much less how it is that these are lost in cave-dwelling arachnids. It's essentially terra incognita. So this is where we come in. To address this question, I'm pursuing this through a binational collaboration with Dr. Efrat Gavish-Regev, a PI at the Hebrew University in Jerusalem. Shown here are various members of our team, including a couple of new people who've just joined the team, Caitlin Baker, Charlotte Kanzler, and Zina Ganem. And to me, comparative biology has to begin by looking at processes in nature. So off we go to the field. Oh, I should add here, there are a couple of other people involved in the project, a couple of undergraduates who absolutely will never listen to what I tell them, and so they absolutely refuse to send me photographs, but rest assured these people exist. Okay, so off we go to the field, and what we're doing here is we're surveying a very large number of cave systems in Israel. So we go into these caves, we're looking for cave arachnids, and we bring along with us a mobile lab to identify different species as well as obtain embryos of these animals. Shown here is a survey of some of the fieldwork we've been doing in the past couple of years. This project really started or took off in about 2017. So in the last couple of years, we've surveyed about 42 cave systems, and several others are planned across Israel. Our quarry then is 3 different groups of lineages, 3 different arachnid orders that exhibit these, what we presume to be sister species pairs of epigean and troglobitic species. So in the top row are spiders of the genus Tegenaria. These are funnel web spiders. In the middle, we have web spiders of the genus Chirinus. And at the bottom, you are looking at the Laniatorid harvestman or daddy longlegs. This is the genus Hasus. Part of this work is steeped in biodiversity discovery. For example, if Gavish Regev's team described Carinus israelensis just 4 years ago, and this species in the bottom right, Hasusna asane, this was described by our team in a work led by Shlomi Aharon, Efrat's PhD student. This is only the second Laniatoris that is known from Israel, and it is found in exactly one cave. It is a point locality endemic. Okay. Why these three systems and what are we doing with them? In this talk, I'll really try to scratch the surface and tell you about a couple of the first insights that we've had. Here I'll address three different topics. The first one is the duplication of these genes something that is specific to spiders, or is this shared across all of the book lung-bearing arachnids? The second, are these duplicates doing anything interesting with respect to eye loss? That is, are we seeing differential expression in blind versus eye-bearing species? And lastly, can we actually go beyond gene expression and obtain functional data linking these gene expression patterns to functional outcomes? So in the interest of time, I'll really focus on one of the three data sets. And this was a data set from the whip spider Carinus. So these animals are not actually spiders. They are a different arachnid order. They don't have venom. They don't spin silk. And one of these species, Ioneticus, does have eyes. The other has a vestigial or almost absent set of eyes. And this work is largely led by my PhD student, Guilherme Gainet, co-led by my postdoctoral fellow, Jesus Ballesteros. So they bring here a combination of skill sets in developmental biology and bioinformatics. What they do then is they go into the field, obtain embryos of these whip spiders at specific stages, and they establish resources for examining gene duplications in Amblypygi. I should point out there are no genomes or developmental resources for these animals. This is something that Guilherme and Jesus are building up from scratch. So shown here in these photographs, these are embryos of Amblypygi, and this is, for example, a couple of different stages of Ioneticus. And this is the eye-bearing species. These tiny brown spots that you're seeing on the screen in the late stage, these are the eyes. So that's the median eyes here. These are the lateral eyes on the sides. And our approach then was first to establish developmental transcriptomes for whip spiders to examine how it is that the RDGN homologs occur. Are they single copy or duplicated? And but I won't dwell on the methods again in the interest of time. Happy to take questions on this afterwards if there are any. Here's what we found out. First things first, what you're seeing here is a summary of our search for the homologs of RDGN genes. Shown here in this tree are, again, the two spiders from which we previously had data. Here are the two whip spiders. So this is another arachnid order for which we just generated data. Centroides is a scorpion. So this is the last major lineage of the arachnopominids. And then Archaea are things like mites and ticks. So arachnids that are outside of the arachnopominids. What we found is that every gene that is duplicated in spiders is in fact also duplicated in at least one of the whip spiders and also in the genome of the scorpions. That is, those duplicates that you saw in the spiders, that's not a spider-specific phenomenon. So having duplications by itself does not explain the diversity of eyes or the specialization of eyes for different purposes. Rather, it's what might be of greater interest is what these genes are doing in these different lineages. Okay, the second question then is, what actually are they doing in these different lineages? And are they differentially expressed in different developmental contexts? So to answer this question, again, using these resources that they built, what Guilherme and Jesus did, they performed a differential gene expression analysis. The way that this works, they take the reads that they obtain from different sequence stages of these whip spiders, and they map them back to the transcriptome. And they're using the distribution of read counts as a proxy for gene expression. The more reads you recover in the short read sequencing, the greater the expression of your gene of interest. Okay, they performed two comparisons. The first one is within, or rather, I should say, between the stages of the species that has eyes. So this is that epigean species. And they again compare two stages right around the time the eyes are forming and then after they've already finished forming. And they surveyed the RDGN genes using this bioinformatic approach. They assessed differential gene expression. What they observed then, and this is summarized in the graph, all the bars that are below the line, below 0, these are highly expressed, or more highly expressed in the stage before eyes are formed. And what we observe is that there's a number of paralogs that are indeed differentially expressed in the pre-eye stage. So they appear to be doing something interesting, whereas their paralog is often not differentially expressed. For example, compare dachshund A to dachshund B. So, okay, that's fine. The second comparison that they perform is between the epigean and the troglomorphic species. So the one that has eyes versus the species that does not. And once again, here, when they perform this comparison, what they observe is that, yes, indeed, there are some genes that are differentially expressed, but comparing the two sets of genes, they are not the same. In other words, yes, these RDGN genes are expressed during development, these paralogs, these duplicates. They appear to be doing different things in different developmental contexts. So this is interesting and it's useful insofar as it gives us a tighter set of candidates to pursue for functional interrogation. In other words, having obtained these expression patterns and these expression data, we want to know what these genes actually do. So how do we pursue this particular question? What is the function of these paralogs? To address this, we interrogate their function in the leading model system for arachnopulmonates, the spider Parasitoda tepidariarum. One of the techniques available for this species is RNAi, RNA interference, or gene silencing. So in this study, what we've done is we've knocked down or silenced 3 of these paralogs: Cine oculus A, or SOA, orthodentical B, and optics B. Right? The approach is we inject females with double-stranded RNA for our gene of interest, which she mates, we obtain lots of cocoons from her, and then we score thousands of embryos for phenotypes. So this is our approach then is once those embryos hatch into these post-embryonic stages that you see here in this video, we attach them to tape and we essentially observe their development. And already you can see the eyes that are present in this stage, and ultimately they'll molt into a first instar. Taking in the kinds of data that we examine, we dissect off the heads, and here again, these half moons are the median eyes, the median ocelli, and these are the lateral eyes on the sides. So again, in the interest of time, I'll just talk about one of those 3 genes, and that's SOA. SOA is expressed in all of the eyes of the spider. What we found, and this is a work that was performed both by our team as well as by Shlomi Aharon, who visited our lab last summer, in this phenotypic analysis, what we observed after scoring thousands of embryos is that knocking down Cine oculus A will result in the loss of all of the eyes in some embryo or another. So it's quite a broad phenotypic spectrum, but we do observe the phenomenon that this gene that is expressed in all of the eyes is responsible for patterning every single eye type. What you are looking at then is the first functional data genetic data point that addresses morphogenesis of any arachnid eye type. So what I told you today is that these duplicates of RDGN genes occur in groups outside of spiders. They occur across the arachnopulmonates. They appear to be doing interesting things in different developmental contexts, and some of them may be responsible for the loss of eyes in cave-dwelling species. And one of them can, for the first time, be linked to a functional outcome. That is, it is indeed responsible for patterning eye types. So today I really just talked about developmental biology and functional genetics. More broadly, the project that we're pursuing then is touching upon biodiversity discovery. That is, describing some of these species from caves for the first time. We're also looking into species delimitation and the biogeography of these different arachnid groups. I didn't really have time to talk about any of that, but suffice it to say, we are looking into some of these other nice components of Israeli cave biology. We don't just harass baby spiders all day, although that is a part of it. So I'm afraid I'm really running out of time. Really quickly, this work was made possible by a really fantastic team of people and various funding sources. My special thanks to the organizers for having me here today. Thank you for listening.
Thank you, Dr. Prashant, for your wonderful talk. We have some questions. The first question is...
Thank you, Dr. Sharma.
How long is the development of the this arachnid egg, and at which time the eye begin to appear?
Sure. So how long is the development? So in Parasiatoda, the development will typically take something like 6.5 to 7 days at room temperature. Or sorry, I should say at 26 degrees Celsius. The eye's primordia are first apparent in the embryo as invaginations that you're seeing in this. Actually, I'm going to share the screen again. Bear with me for 1 second.
I'm sorry.
Okay, so the these eye primordia start to develop around stage 10.5 or 10.2, as we call it. They will really first begin to develop closer to when the embryo is about to hatch. Now, that's the case for spiders. In whip spiders, the development is much longer. In some species, it can take up to 80 days. So here, this is why we really have to combine field biology with developmental genetics. We can't really raise these animals in the lab very successfully because they are tricky to work with. They have very long developmental periods. That being said, and I should point out, one of my students, again, Guilherme Gainet, is actually developing amblypygy as a model system. He's actually figured out how to take a different species, this is a Floridian species, raise it in the lab, pull embryos off the female, and he's pioneering developmental gene expression techniques for amblypygy for the first time. So the answer is they're very disparate. They're really different from each other in terms of development and when the eyes form, but they tend to form fairly late in development.
We go the next questions. Based on the known species with lost age, fish, isopods, etc., is there any part of the gene network more sensitive to be affected?
Are there any parts of the network that are more affected in eye loss? So the short answer is it does seem like modifications of the RDGN genes, so this upper hierarchy, this cascade of genes that sets up the eye, it's modifications of one or more of those members that is typically responsible for eye loss. This is why it's among our candidate genes. So we're prioritizing these because we think that might be where the answer lies. What generally speaking has been shown again, this is addressing morphological convergence more broadly, it's sometimes the case that morphological convergence is a result of parallelism in gene networks. For example, in stickleback research, it's been shown that the loss of the armor when the marine lineages invade fresh water, it's the result of repeated selection on the pit X locus. It's in fact the same enhancer of the same gene is repeatedly targeted for selection when different lineages lose a structure. Is that broadly the case? Is that generally true, even for example, for troglomorphic arachnids? We don't know. So this is why we want to look at 3 different orders at the same time in the same biogeographic theater and in the same time frame for eye loss to investigate, is that true? Does morphological convergence require convergence of gene networks?
Okay, thank you, Doctor. No, sorry, Ale, we don't have any time.
Oh,
You
okay.
can type the rest of the question. You can type it in the Q&A. But thank you very much. That was a wonderful talk.
Our next speaker is Catalina Pinto. She is in paleontological biology working in Swansea University in the UK. Her research focused macroecological dynamic with the special emphasis on extinction. We'll talk about giant shark. Thank you, Catalina.
Thank you. Can you all hear me?
Yes.
Okay. And can you see my slides?
Now I can.
Yeah, okay, cool.
Yeah.
Okay, so I'm going to be talking about how to be a gigantic shark. And before I start, I would like to acknowledge my collaborators on this work, Juan Cantalapiedra, Ken Shushimada, Daniel Field, and Jerome Mars. And before I start talking about how to become a giant shark, how to be a giant shark, I want to start by talking about how did I get to this point of being interested in this question. And it was when I started looking for megalodon teeth in Panama. So Megalodon is the biggest shark that had ever lived. It could reach up to 18 meters of total length. So it was massive. It was like a transmillennium. And when I was doing my master's in the University of Florida, part of my work was to dig for fossils in Panamanian sediments of the Miocene. And of course, we collected a lot of different species, but obviously the teeth of Megalodon was one that we found was fascinating, as many other paleontologists, because they are so big and also because it's extinct. So the first thing that we discovered by looking at these teeth from Panama is that they seem to be very small compared to what we have seen in other locations or in museum collections. So we hypothesized that these teeth belong to babies and that Panama was a nursery area based on the information that we had from recent species of sharks. So we went ahead and collected as many shark teeth as we could of Megalodon, and we measured them and we calculated the body size. And we did that by extrapolating the relationship that exists between tooth size and body size in the great white shark, which is the modern analog of Megalodon. And we used this extrapolation and we found out that most of the individuals that lived in Panama 10 million years ago were juveniles. Some of them were babies. And then we proposed that Panama was a nursery. So a nursery means that big Megalodon moms will go to this area and give birth to their babies there, and they will find enough food and protection to grow and then go to other places. So after we had this proposed this hypothesis, we became even more interested on this species, particularly because it is extinct. So the next thing that we wanted to find out was when it went extinct. At the time, around 2015, there were some documentaries in the Discovery Channel proposing or suggesting that the species was not extinct. So we took advantage of this momentum to put together this very quick paper where we analyzed the most recent records of Megalodon. So in this graph, all these blue lines represent all the most recent records, and there are lines and dots because in the fossil record, you have range of ages, so from 10 to 5 million years, as opposed to a punctual or a absolute date. So we use these dates and we use a mathematical model, and we found out that the most likely time of extinction for this species was between 2 or 1, around 2 million years ago. So yeah, it's very much extinct, and it went extinct even before the evolution of humans 2 million years ago. And we also noticed that the evolution of giant teeth, I mean whales and cetaceans, took place after this. So then once we knew the most likely time of extinction of the species, we wanted to test the hypothesis of climate change as being a factor that causes extinction. So the next question was how or why it became extinct. And like I said, it had been proposed that the water had become too cold for this species that prefer warm waters, and that's why it became extinct. So we did this niche modeling analysis, and we found that actually Megalodon was not affected, or the distribution of Megalodon was not affected by temperature. It could live in different temperatures all around the world, and even after the different climatic changes that occur after this extinction, in terms of temperature, it could have lived all around the world. So temperature was not an important determinant of the extinction. Instead, we found that there was some coincidental timing with the time of extinction or the decline of the species in terms of occupancy or abundance, and in terms of the distribution, with the appearance of new competitors like the modern great white shark or the modern orca, and also the decline of potential prey. So at the time, a lot of prey like big mammals that Megalodon would have feed on also declined. Some of them became extinct. So then is when we started thinking about understanding the extinction of this species better by looking at the whole marine megafauna. So we wanted to see if Megalodon became extinct on its own alone, it was just one extinction event, an extinction of one species, or if instead there was an extinction event and Megalodon went extinct along with many other big animals. So we gathered all the records of the marine megafauna, so everything that was big in the oceans, sharks, mammals, seabirds, sea turtles, and so on. And we assessed extinction rates, and we found that actually at the time of the extinction of Megalodon, for the entire marine megafauna, there were very high extinction rates. So there was an extinction event in which at least 36% of the genera became extinct. So it was an important extinction event, even though it wasn't a mass extinction event, it was very significant. So Megalodon became extinct with other animals that were big as well. And then we assessed what were the potential intrinsic traits that were correlated with this extinction. So we tested for body size, diet, vertical position, habitat, and thermoregulation. Thermoregulation being the capability of animals to regulate their body temperatures. So we have in the marine megafauna, you have endothermic animals like all the mammals and the seabirds. And you have the mesotherms that are animals like some sharks and some sea turtles that are able to regulate at some extent their body temperature. So in the case of sharks, for example, the great white shark, they're able to regulate the temperature of their brains and their stomachs. So that's mesothermy. So those animals that were either endothermic or mesothermic were more likely to go extinct. And we also found that around the time of this extinction, there was a significant decline in the area available for these animals. And that's the graph I'm showing here. Oh, my cursor is not showing, but the graph at the right side that is showing how area was increasing and decreasing, and it's a massive decline in area in the late Pliocene. So because these animals that are endothermic or mesothermic, homeothermic animals, need to eat a lot so they can regulate their body temperatures, we proposed that this was a main driver of extinction. So I became very interested in thermoregulation because we had proposed that it had a role in the extinction of the biggest shark that ever lived, but also simultaneously there had been other papers or other authors that had proposed that it was also involved in the evolution of giantism. So we became wondering, well, if this particular trait of being a mesothermic shark can be potentially can drive you to extinction in a way or another, or can be correlated with extinction, how come it also helps you to become a giant? So then is when how giantism is achieved, that question came up. So to assess this question, you have to define giantism, and there's of course different concepts and different it has been proposed to use it in different ways. So we went through the literature, looked at all the potential definitions of giantism and apply it to sharks. So combining all the different definitions, we set up like an arbitrary cutoff for what is to be a giant in terms of sharks. And we said it's going to be more than 6 meters. So let's call giants everything that has more than 6 meters of total length. So if you look at the diversity of sharks, both living and fossil, there are around 14 giants. So 14 species that have reached more than 6 meters of length. So you have the super giants like the whale shark, which is the biggest fish today that we still have in our oceans, and you have the extinct megalodon. But also you have the basking shark, the manta ray, the tiger shark, the great white, the hammerhead, Greenland, megamouth, goblin, and a number of extinct sharks that had been estimated to be more than 6 meters. Interestingly, we noticed that some of these sharks that were giant were also not normal in a sense of feeding and thermoregulation. Not normal in the sense that there are around more than 1,000 species of sharks, but only 13 or 14 are filter feeders. So most sharks, or the normal sharks, are macrophagus, they eat big things, and just 14 species are filter feeders, and a lot of these are giants. So the whale shark or the basking shark, they are filter feeders. And also some of these giants were also mesothermic. So being able to regulate their body, the temperatures of their body is only achieved by a handful of species, like the great white shark and some members of the same family. So it was very interesting to note that for the group of giants, we have these outliers of these very specialized sharks. And that brought us to think about the role of trophic label, because in fish, it had been proposed before and is widely accepted in the literature that there is a strong correlation between trophic level and body size. So in that sense, you would expect that the animals that feed in the very high, in the highest parts of the trophic chain, then they will become giants. So if you're a big predator, then you're going to be a big animal. So yeah, an apex predator like the great white shark, or in this case megalodon, would be the biggest one. But we also know that we have filter feeders that are also giant, but they feed very low in the trophic chain, so they eat plankton. So what is the role of this trophic level in giantism? So what we did was to gather a tree of sharks and rays from the literature. My collaborator Ken Shushimara helped us with taking that tree and adding fossils. So we added some fossil clades that we knew, and there were not many of them because the relationships of the phylogenetic relationships of extinct sharks are not well known, and in a lot of cases we haven't identified them to the species level. So we don't have a lot of fossil sharks to add to this tree, but we did the best we could with the information at hand. So we added these fossils, and at the same time we assigned these traits to the tree. So total length, so how big the shark was, or they are, trophic level, so either if they're predators or filter feeders, or where in the food chain do they eat, the feeding mechanism, and the thermoregulation. We gathered this data we downloaded this data from FishBase. Then Jerome and Juan helped us with mapping these traits in the phylogenetic tree of sharks. So I'm not going to dig into or I'm not going to spend a lot of time explaining the methods, but basically we did phylogenetic regressions, we assess ancestral states, and do evolutionary assess for evolutionary rates. So we took this information, and this is what we found out. So as we expected, because it's the case with fish, trophic level and body size are strongly correlated. However, mesotherms and filter feeders deviate from this relationship. That means that they don't quite fit in this relationship. Not only that, but also mesothermic and filter feeders are significantly larger than the rest of sharks. So as you can see here, we have a histogram of the distribution of sizes of sharks. And these group of species, the red and the purple, have significantly bigger sizes, have evolved bigger sizes than the rest of the sharks. Then when we mapped all of these traits, we looked at how they have evolved in the tree of life for sharks. So we found that mesothermy evolved once, a long time ago, around 140 million years ago, and is the triangle that you see here in the tree. We run the same analysis, but we excluded the fossils, and we found that if you don't include fossils, then mesothermy could have evolved 3 times in the Cenozoic. But for the rest of the talk and for the sake of the paper, we did all our interpretations based on the results we got with the fossils. When we looked at filter feeding, we found that it evolved 4 times in 4 different clades, and most of them were around 50 million years ago, although one of the dates was older than that, 80 million years ago. This pretty much corresponds to what has been proposed before for filter feeding. Now, when we looked at the ancestral states of the different clades that were gigantic, we noted that most of the clades that have gigantic sharks have a very big or a big ancestral state. So they descended from a big ancestor, much bigger than you would have for the clades that don't have giants. And interestingly, also there was one clade, the labniforms, which is the order of sharks in which the gray white and megalodon belong to. It shifted its mode of evolution. So that's the red part of this tree here in the right. And this clade deviated from the evolution of body size that sharks were having. This clade showed a deviation, and by that, then what happened is that it had a different optimum size. Then the first giants evolved, and then there was a diversification of species within this clade. So this clade of labniforms is very important for the evolution of giantism. You have mesothermy, you have giants, you have filter feeders. So it's a very important place for the evolution of giantism in sharks. So when you take everything together, you can propose, or we propose different mechanisms or pathways to become a giant shark. So that's why it's how to become a giant shark. So you have these three ways. One is by becoming a mesothermic. Another way is by becoming a filter feeder. And another way is don't do any of those things, but you won't become a super giant. You only be bigger than 6 meters, but never reach 18 meters like with the other pathways. So for the mesothermic pathway, like I said, there's a shift in body size regime. You reach an optimum size of 4 meters, then you evolve mesothermy, and then the first giant appear around 100 million years ago, and then there was a diversification. And around 23 million years ago, there's the evolution of the biggest mesothermic shark, which was Megalodon, but it became extinct around 2 million years ago. Then if you're a filter feeder, there are multiple appearances of filter feeding. You develop that, and then you have the biggest shark that we have today, which is the whale shark that still lives in our oceans today. So the first giants that appear in the mesothermic pathway and the filter feeding pathway, they appear during times of climatic changes. So in the case of mesothermy, it was when there was global cooling and a lot of predation. And in the case of the whale shark or the filter feeders, it was when there's a lot of productivity and a lot of predation as well, and also niches that were available. So we proposed that these specializations were a response to enhance prey intake. So if you're a mesothermic and there's a lot of global cooling and predation and you develop mesothermy, then you're able to expand your niche. You can live in cold waters because you can regulate the body temperature. And also that mesothermy has been proven empirically to make your predation more efficient because you can swim faster and predate with more efficacy. And in the case of filter feeding, well, if there's a lot of productivity, obviously you want to take the best advantage of that by eating plankton, because plankton is very abundant when there's a lot of productivity. So all of these specializations were a response to enhance prey intake. However, giants are unfortunately under threat. So in the case, as I said, all of these specializations were a response to climatic changes, and they depend on quality and abundance of food. So the only reason why we have the whale shark today is because there's a lot of plankton in the oceans and they can feed of this plankton and then be very large. And in the case of the megalodon, well, there were a lot of prey available for it. But in the case of mesothermy and in the case of megalodon, when areas were reduced during the Pliocene due to the different climatic changes like sea level changes, then there was not enough area to find enough prey and these species became extinct. The other path where filter feeding still persists today because plankton is more abundant and it's easier to find. However, today we could say that it's also in threat because there are a lot of microplastics and toxic microplastics in the oceans, who's now threatening this very important aspect that is needed for the maintenance of giantism in our oceans. And with that, I would like to finish by thanking you all for listening to me, and also obviously my collaborators and all the funding agencies that made this work possible. And with this, I'll take any questions now.
Thank you, Dr. Catalina. Again, we thank you for your participation. You give an excellent talk. Have one question. Do larger mammals present similar macroevolutionary patterns to shark?
I'm not an expert on mammals, but I can say that in terms of filter feeding, yes, it seems like the way that giantism in marine mammals evolved was also a response to a lot of productivity in the oceans, and because there was so much plankton available that allowed marine mammals to also evolve gigantic sizes. So they seem to have a similar kind of pathway there. Yeah.
Thank you very much. One second questions is, do you think megalodon have actual parent care?
No, I don't think so. We don't see that in sharks today. Definitely, we don't see that in modern relatives of megalodons. So I don't think, however, using a nursery or yeah, using that habitat as a nursery could be in a way parenting care, because you know, you're leaving your babies in a place where they're going to be protected. But other than that, no.
Thank you, Catalina. I haven't more time. Continue, Jime.
Ok, gracias Catalina, fue un honor escucharte, me encantó tu charla y si hay un par de preguntas más puedes resolverlas ahí escribiendo por el icono de QIA. So she will answer the question typing there.
Okay.
So thanks.
Of course, thank you guys.
We are going to move forward. Our last speaker is Dr. Claire Fabre. I am really honored to present her. She's working in the London Natural History Museum, and she will talk today about metamorphosis in salamander. She will speak and receive question and answer in English. And so, Dr. Fabre, you can share your screen now.
Okay, thank you. So, can you see the pangolin? Can you see my talk now?
Yes, I can.
Okay, great. So, hi everybody. And first of all, I would like to thank the organizing committee of the SBE for inviting me to give a talk in this symposium. So today I will talk about the results that I recently published in Nature, Ecology and Evolution, which are about metamorphosis as a driver of morphological diversity in salamander. So when you think about it, metamorphosis is an incredible developmental process. It implies an abrupt change in the morphology and ecology of an animal during its lifetime. So with its transformation from a larva into an adult. As you can see here, for example, in frogs or in butterfly. Okay, I will point out now. So what is really incredible is that for a same individual, different morphological stages are produced with the same genetic material. So metamorphosis is really widespread in the animal kingdom. It's nearly 80% of insect species and at least 1/2 of the non-vertebrates that are undergoing metamorphosis. You can see on this slide some example of some species with their larval stage and the adult stage. So if we take a closer look at the vertebrate, here you can see a phylogeny of vertebrate at the genus level. In red, you can see species that are encountering metamorphosis, so which have a complex or an indirect kind of life cycle. And in yellow, you can see the species that have a direct kind of development, so a single life cycle type. And if you look at the number of species right under the name every time of the taxa, what you can see is that nearly all the most specious lineage of vertebrates have a complex life cycle. So this may indicate that metamorphosis may be a really important driver of biodiversity. So the benefits of metamorphosis are not really obvious, and several studies have tried to understand the advantage of such complex life cycle. So it seems that metamorphosis comes at a cost, as the animal is particularly vulnerable during metamorphosis, and there is a high risk of predation. Furthermore, it is costly in energy, as animals often show an overall morphological repatterning, which is associated with drastic changes in physiology, diet, and habitat use. On the other side, despite all these costs, there are several benefits to be a metamorphic species. So the first advantage is that you do not have to share the same resource with the adult. And the second advantage is that the intraspecific competition between the larval and the adult stage is reduced, which may favor the population growth. However, we still do not understand how different ontogenetic stages appeared, why they were maintained through time, and how they impacted species diversity. So it's in this context that we decided to realize this study. And in this paper, we were doing a focus on salamander. Why? So salamander is a really interesting group because they display a tremendous diversity of life cycle. So some of them are what we call biphasic, and they are exploiting different environments during their morphologically different life history stages. So they hatch from an egg on the larval stage that is living in the water and feeding in water. Then they encounter metamorphosis and end up as a terrestrial adults that have a morphology that is totally different, ecology too, and diet. Other species are what we call direct developing species. And these species hatch directly as terrestrial phenotype, and they lose their larval structure and ontogenetic repatterning. So what is important here to see is that they stay during their entire life in the same kind of environments, mostly terrestrial environments. And another thing that is interesting to notice is that they still encounter metamorphosis, but in the eggs. Then other species are called phelomorphic, and these species keep larval traits even at the adult stage, even if they are reproductively active. So they hatch in the water to a larval stage, and they mainly grow and lost a little bit some of their larval traits, and they stay during their entire life in the aquatic environment, still feeding the same kind of thing. So as you can see on this phylogeny that is coming from a paper from Ronald Bonnet and a collaborator in PNS, you can see that salamanders include 10 families, which display different kinds of developmental strategies that appear independently during their evolution. So when you look at that, you can see that in red, it's biphasic, and biphasic is an ancestral condition. And you can see in blue there, for example, that there is several independent evolution to phelomorphy. And you can see here in yellow that you've got one appearance of direct developing strategy. And inside this family that is called Pletodontid or langless salamander, you can see that you have a re-evolution of biphasic species and phelomorphic species. Then for this study, what we wanted to do was really to test the impact of metamorphosis on cranial shape evolution. Here is just a representation with some picture of salamander that are representing the external morphology diversity. And what you can see that's really striking is that even among the same genera, such as, for example, the Eurycea, you've got this different kind of morphology, with this one that is phelomorphic and this one that is biphasic and living in terrestrial environment. You can see here the famous giant salamander in Japan, here the fire salamander that you can find in Europe, and here, for example, a tiger salamander that is really closely related to the axolotls. When you look at the inside, so the bony part of the head, when you look at the cranium, you can see that the shapes are really, really different. And it was kind of tricky to work on that, but funny at the same time, because you've got a lot of present absence of the bone. And yeah, I'm used to work on mammals, and I can tell you mammals are way easier to work with because most of their bones are still present. So, as I was saying, we wanted to test the impact of metamorphosis on cranial shape evolution, and we had some prediction. So the first one is coming from the study of Ronald Bonnet and his collaborator in PNS. So they were looking at the body shape and vertebral column evolution in salamander in larval and adult stages. And they show that species that have a simple life cycle, so the direct developing species and phelomorphic species, tend to have a higher rate of evolution than species that are biphasic. Then, on the other side, we predict, and these predictions are our prediction this time, that species that have a complex life cycle, so the species that are biphasic, with a change in diet and environment, tend to be more modular than the species that have a less complex life cycle, such as the phelomorphic species or the direct developing species that are staying during their entire life in the same kind of environment. And finally, we predict that the bones that are involved in food processing or those that are remodeled during metamorphosis will have an elevated morphological diversity and an elevated rate of evolution, and thus and increased ability to evolve. Our dataset was composed of 148 species, so it's only CT scan. And you can see here on the backbone phylogeny, at the level of the family, the number of species under brackets for each family. It's a really good representation of salamanders, and especially of their different kind of life cycle and their diversity. What we did some shape quantification using geometric morphometrics. You can see here a lot of sliding semi-landmarks to represent accurately the shape of the bone, and the different kind of color are representing each bone. So let's move on to the result. The first thing that we were doing were exploring the morphological difference depending on life cycle. So to do that, we were testing using a phylogenetic MANOVA if there was any kind of cranial shape difference depending on life cycle, and yes, it's the case. And we were running a principal component analysis. And what you can see here is a really nice clustering of the species depending on life cycle. So you can see in blue the pedomorphic species here, in red the biphasic species, and in yellow the direct developing species. So the first principal components tend to differentiate species with a fully aquatic lifestyle in comparison to species that are spending all their time or part of their time in terrestrial environment. Whereas the second principal component axis describes difference between species with an aquatic and terrestrial life cycle and with fully terrestrial direct developing species that are the faisceau-dense. Other kind of things that we can see on this morphospace is that cranial shape vary depending on feeding modes too, with the species that are using jaw prehension, such as here, the other ones that are using section feeding, and these ones that have a ballistic tongue, such as the chameleon. If you look at the shape change, you can see that the pedomorphic are there with a really streamlined cranium, which is important for moving and feeding in water, whereas the more terrestrial one have a more robust skull like that. Oh, just there. Okay. Then we were testing for difference in morphological diversity depending on life cycle, and we show that cranial diversity is influenced by life cycle, with the pedomorphic species in blue again that have the higher disparity, whereas the direct developing species in yellow have the lower one, and the biphasic tend to be intermediate, but still with a low disparity. Then we estimated the rate of cranial evolution on the chordata phylogeny, so on the salamander phylogeny. And what you can see here is that the color gradients on the branch indicate the rate of shape evolution, with warmer colors that are corresponding to a higher rate of evolution, and cooler colors that are corresponding to the slower rate of cranial evolution. The gray triangle are corresponding to the shift in rate. And what is interesting to see here is that every time that there is a transition from one life cycle to another, you can see a shift in rate. And another thing that is nice to notice is that pedomorphic species tend to have a higher rate of evolution, whereas direct developing species tend to have a really low rate of evolution, and biphasic species tend to have an intermediate rate of evolution. So if you remember the predictions that we were saying at the beginning, we were predicting that direct developing species and pedomorphic species will have a higher rate. And in this case, so we are looking at a different kind of traits, and we can see that pedomorphic have a high rate, but it's not the case for the direct developing species. So from one trait to another, we may find different kind of things. Then finally, what we were doing, we were testing for the modularity and integration depending on life cycle in order to test if species that are biphasic with a change of diet and environment during their life will be more modular than species with a simple life cycle. So the concept of modularity is interesting. I don't know if you heard about it, but it's a way that some part can evolve quasi-autonomously from each other. And modularity is often linked to evolvability, at least in the population level, because you cannot look at that at macroevolutionary level. And having a a bigger evolvability, can then give you the ability to adapt quickly to a changing environment and so on. So it's a kind of a nice concept. So here, what we were able to see is that the species that have a complete metamorphosis are more modular than the pedomorphic ones. So you can see here that you've got 11 modules, and here you've got more modules. And you can see here a network that is just representing the correlation inside the structure. So bigger the circle is, bigger the correlation inside the structure is, and bolder the line are, and stronger the correlation between two structures are. And what you can see here is that, for example, in biphasic and direct developing species, you've got modules for the maxilla and the premaxilla, but also for what we call the suspensorium, which is composed of the squamosal, the pterygoid, and the quadrate. And it's basically where the jaw is inserting. And yeah, what is nice is that we've got a signal for metamorphosis here and not the change of environment. What we were looking at then, we were testing the tendency to evolve disparate morphologies depending on life cycle. And to do that, we were assessing the relationship between the rate of evolution and the disparity for each module. So we were looking at the correlation between rate and disparity for each module. And what you can see here is that our results show a correlation between the rate and disparity only in completely metamorphic species, so in biphasic and in direct developing species. So here we show that the different parts of the skull don't always evolve in the same way. And for example, in species that undergo the complete metamorphosis, some modules have a higher disparity and a faster rate, such as for the suspensorium and the maxilla. And these modules, interestingly, are the ones that are implied in feeding strategies that are supposed to change after life cycle, during life cycle after metamorphosis. But it's still kept in direct developing species. That's really interesting. So to conclude, what we can say is that we found shape difference depending on life cycle. There is a higher disparity and rate of evolution for pedomorphic species for each bone. So species with direct development have the lowest disparity and rate of evolution. We found a shift in cranial rate depending on change in developmental strategy. Direct developing species and biphasic species are more modular than pedomorphic ones. So we were not expecting that at all for the direct developing species. And then the structures that are implied in feeding have a higher rate and disparity in metamorphic species. The structure remodeled during metamorphosis are less constrained than other bones in metamorphic species. And finally, we can conclude that metamorphosis has driven cranial shape diversification in salamander, but it will be nice to go further and to include now the larval stage. So it's what we plan to do in the future. And yeah, we will see if the larval stage tend to fall in the same kind of morphospace than the adult stage. Can be nice. And to finish this talk, I really want to thank all the co-authors of this paper, again, the organizing committee of the SBE, the collection, and the Goswami Lab. And if you have any question, I'm really happy to reply to them.
Okay, thank you very much, Anne-Claire, for this great talk and the great work you did. And we're running out of time, but we have a couple of questions that we may say here. The first one, are these patterns repeated in other vertebrates with these complex life cycles?
So some people were studying that in frogs, for example, and it's a little bit more complicated because during metamorphosis in frog, you've got an overall repatterning. It's the cranium of the frog of the larval stage and the adult stage are really different. So it's more difficult to see. And from what we're finding, some collaborators and other people, if you just look at the adult stage, it's not as straightforward. So in salamander, it's just a partial repatterning during metamorphosis. You only got 2 bones that are repatterned. All the other ones are out there and appearing at the larval stages.
Okay.
So yeah, and for other species, I have no idea because most of the time it's not studied in this way. And for example, in insects, it's pretty complicated because finding homology between what you've got in the larval stage and the adult stage, it's kind of tricky.
Okay, well, we have time for one more question, which is, do you know of any other trait that may interplay with metamorphosis to produce the macroevolutionary dynamics observed in salamanders?
Trait or ecological factor?
I don't know, it says trait.
Trait. So I suppose other trait, it's complicated. I have no idea. Other trait, I suppose that different part of the body will respond differently to that. But yeah, for other factor, I could have replied, but for other trait, it's kind of tricky because most of the people were studying mainly the external morphology. And it's mainly body length and things like that. So,
well, no worry. The discussion can keep going on Discord. So any question you have and any clarification you want to do, you can keep going on Discord. Now, just remember you that we have the Symposium of Gender and Diversity in STEM, which is the last talk of the symposium. And we have put the link in the chat. And that's all for now. And thank you very much for being here to all the participants, to all the panelists. And it was great. And it was such a great time to have you here. Thank you very much.
Thank you very much. We love you here.
Thank you.